Over to you, Vikraman. Ready.
Good afternoon, everybody, and welcome to the last of the webinars in our series for the Amrit Grand Challenge or Jan Care program. The Amrit Grand Challenge Jan Care program is a program of BIRAC in partnership with IKP, NASCOM, MIT, and many other partners. We aim to identify 75 health tech innovations and fund them to mark the 75th year of independence. We will be funding 60 early stage innovations to the tune of up to INR 10 lakhs each. 13 late stage innovations will get an investment of up to INR 20 lakhs each, and 2 advanced stage innovations will get funding of up to INR 50 lakhs each. The call for applications is open. This is a webinar to invite applications. You can see the URL to apply below on the slide. Our team will also post the link in the chat box, and you may log into the BIRAC website and apply. Only online applications will be accepted. The last date for applying is March 31st, 2022. So once your applications come in, there will be an online screening process, and then there will be a presentation stage, and hopefully we will be able to identify and lock down these 75 innovators to be announced by the highest authority in the country on August 15th, 2022. We also have a lot of industry partners who have come in to help these startups and innovators. We have support from AWS. Each of the teams will get cloud credits of up to $10,000. This is over and above the funding that we offer. We also have support from MathWorks, who have offered MATLAB licenses to the companies. We are also in talks with leading pharma companies and medical device companies to come in as funding partners and as mentorship partners to enable these entrepreneurs to go forward in their journey. The primary theme of the call is digital health, and across that we have 6 topics, which is access to primary healthcare in tier 2, tier 3 cities, solutions to enhance patient compliance, health data collection, predictive analysis, data privacy, storage, and security, solutions for improved community outreach, and data-driven modeling to enable pharma, biopharma research, development, and innovation. Today, our webinar focuses on the 6th topic, and we have 4 eminent speakers who have come forward to talk about their journeys, the work that they're doing, and hopefully excite you to apply for the Amrit Grand Challenge Jan Care program. With that, I hand it over to our startup mentor, Ranajit Sen, who's also the CEO at Graphene Med and was the former head of business development at Shanta Biotech, to welcome the speakers for our webinar. Over to you, Ranajit.
Thank you, Vikraman. Allow me the pleasure to also introduce Vikraman. He is the CEO of IKP Eden and Senior Vice President IKP. So we have a lineup of brilliant speakers today to address the topic of data-driven modeling in pharma, biopharma, innovation, and innovation and R&D. I would like to introduce to you the first speaker, Dr. Mahesh Iyer, a friend. Dr. Mahesh Iyer is currently the Vice President of Paraxel, a leading CRO. He's the Vice President looking after innovation and technology. Mahesh current responsibilities include adoption and operationalization of innovations and new technologies at Paraxel. This includes a wide array of focus areas, which are decentralized trials, risk-based quality management, real-world data, real-world evidence, creating DM workbench platforms, and expanding the use of AI and NLP in pharma, and so on. Prior to the current role at Paraxel, Mahesh has worked in the pharma space, including BI, Bristol Myers Squibb, and Novartis for over 22 years, mainly in the statistics and programming space, and helping to bring over 10 products to the market. Mahesh was also co-founder of consultancy company, as well as helping to set up the MedTech Incubator Accelerator at Triple IT Hyderabad. In addition to serving as a mentor for many health tech startups across the country, Mahesh has a Mahesh has a doctoral from in statistics from Temple University in Philadelphia. Over to you, Mahesh.
Thank you so much, Ranaji. Thank you very much. I saw Vishu is on the call as well. So good to see you again, Vishu. This is a great opportunity, everybody. And so thank you all for giving me this time to speak to you guys a little bit around data modeling in drug development. So what I'm going to do is I'm just going to focus on maybe 2 or 3 kinds of use cases. And, I'm going to try and stay away from why things don't work today, right? I'm going to try and focus on what currently being done, what are some of these areas. And I've always found that in talks such as these, it's always better to see if there are any questions that I can help address rather than just coming here and giving you a spiel on what we currently do. And so therefore, what I'm going to do is I'm going to leave it open. I have a few minutes at the end for questions. Yeah. If you have any questions while I'm speaking, again, please feel free to stop and or into the chat box, and then let's make this more interactive. So with that, I will. Yep.
I think we lost you, Mahesh.
Yes, Ranajit, I believe we have lost connection, Dr. Mahesh Iyer.
Hello, Mahesh, we've lost you. Okay, okay. He's just trying to reconnect, Vikraman.
Thank you, Ranajit.
While we are waiting for Dr. Mahesh Iyer, uh, I would request the audience, members of the audience, to put up their questions in the chat box. Welcome, shall we move on to the next speaker or wait for Mahesh?
Let us wait a couple of minutes. Maybe in the meantime, if the participants have any questions regarding the funding program, they may push that. I think Dr. Mahesh Iyer has joined. Yes, we have Dr. Mahesh back on the call. Over to you, sir.
Yeah, you guys hear me now, right?
Yes, sir, we can hear you.
Okay. Okay, you guys can hear me?
Yes, we can, Mahesh.
Okay, perfect. Thank you. Sorry about that. I think there was something wrong with my internet connection. So I'm on my using my hotspot from my phone. So that's why I turned my video off. But just do ping me or let me know if suddenly go off again. So yes, I was saying that I don't know where I dropped off, but I was trying to say that I wanted this to be as interactive as possible. So whenever you have any questions or anything, please feel free to just type it into the chat window or stop me and let's have more of a discussion than a presentation from my end. So I just want to start with a disclaimer that all the opinions are mine and do not reflect the views of my current employer or any organization that I'm affiliated with. Yeah. So as I said, what I'm going to do is I'm going to take maybe 2 or 3 examples given the as you might have guessed from the name, the discovery phase is essentially where we try to figure out, whether drug, where we try to discover the drug or create a drug, right, from existing compounds. And here, on the left here, are all the different kinds of activities. Let's say for diabetes, is that you keep your glucose in control, your diabetes is going to be fine. Yeah. For cardiovascular diseases, blood pressure, and so on. Yeah. So we want to.
So Dr. Mahesh, I'm sorry you're breaking. Voice is breaking.
Identify one cure.
Mahesh, your voice is breaking.
Okay, can you guys hear me fine? Better now?
Yeah, it's better.
Okay, good. All right. So as I was saying, so targets, right? So targets are glucose in diabetes, blood pressure and cardiovascular, so on and so forth. So these are all our targets. So we're going to identify the target, validate the target. We want to make sure that we optimize the lead. We want to do all the the modeling for the whole discovery component. We want to analyze the pathway. Pathways are when you start with from a protein, move on to a gene, move on to different aspects. So that's a particular pathway, for example, and then enrich that particular pathway. All of these fall under the big bucket called computational biology. Yeah, a lot of this was done using actual experiments before. So the way we would optimize a lead, for example, was to say, supposing we had 3 or 4 different compounds, we would use all those 3 or 4 different compounds, scan them across a number of different types of targets, and then try to see which one of those 3 or 4 compounds did the best, in identifying a particular kind of in addressing or fighting against a particular kind of a cancer or a particular kind of tuberculosis or whatever it may be. Yeah. Now, what we have started to do is that most of this actually does not happen in the lab, but happens in silico, right? So we actually run models to do most of this work. Yeah. On the other side, you have the computational chemistry bit. Yeah. Is, how do you bind to the ligand, right? So if you have a drug, the drug has to bind. How does the binding happen? Can we do scaffold analysis? How do we figure out whether, what is another compound that is structurally similar to the lead compound? The lead compound may have certain undesirable side effects. So how can we identify another compound which is similar? So how do we figure out structural similarity? If our drug has to go and dock against a particular molecule, then how do we analyze that whole docking system, and all of those sorts of things? All this work, which is the chemistry part, is also done now in silico. So models based stuff to do all of the computational chemistry bit. Yeah. And of late, one of the things that's really working well for us is this whole machine learning bit, right? How are we able to use machine learning to really drive efficiencies in both the computational biology and the computational chemistry? Text mining, can we go and search all published literature where people have already talked about how proteins are binding, how a particular drug is binding to a particular protein? Can we already get that information? All of that can potentially happen through text mining. Yeah? So this is where a lot of the current work is happening on the discovery side from a modeling perspective. Yeah. So if a further detailed diagram for this is what we call the QSAR workflow, which is called the quantitative structure activity relationship. So we start to compile our data sets, either using public databases or literature databases. We basically compile the data sets and identify which is a subset data that we want to investigate, start to process the data, all the stuff, feature selection, normalizing, balancing, and then splitting them into 2080 or 3070 to create test and validation data sets. Then we start constructing our model, whether it's learning algorithm, QSAR modeling, using statistical measures to figure out how robust our model is, and then finally moving into the model evaluation bit. And this is something we do whenever we have to come up with a new drug for a new disease area. Yeah. We also do something very similar when we want to do drug repurposing. Yeah. So you have an existing drug, but you want to use it for a different indication. Yeah. And that's where we start to take all the data that we currently have, any of the omics data, so proteomics, genomics, and so on and so forth. Okay. Get the disease-centric data, get literature data, and then start to generate our computer models, whether it's SVMs, whether it's logistic regression, whether it's random forest, whether it's deep learning. We start to use these models to say, will this existing drug also work on a new indication? Generate a proof of concept, cross-validate, and once we are able to generate this hypothesis and have a large amount of confidence that this new drug, this older drug is going to also hit certain pathways, and so therefore will also work in some of the newer disease areas, then we move into the actual clinical trials bit. Yeah. So these are kind of two areas, where you can see how modeling is currently used in the discoveries part of the drug development. Any questions on this so far?
Yeah, there is one question here, Mahesh.
Yeah.
Regarding structural functional relationship, it is already be available for a particular drug, which you said could be repurposed.
Correct.
So how do you take it forward from there?
Right. So what happens, here is basically, let us say that you have a particular drug, right? And you already know that it is hitting a particular set of, proteins. Yeah, let us say. Now, what happens is that you start to see a lot of literature, new literature coming out saying that, oh, by the way, this protein or this particular pathway is also lit up, for these other disease areas. Yeah. And so you start to say, hey, you know what? Now, for example, I think one of the examples is what is happening, or a lot of false alarms that we had, where we, with the whole vaccine stuff, right? So when COVID came about, there were a whole bunch of existing drugs, that work, let's say in tuberculosis and other areas. And now all of a sudden, people started to think that they were also going to work, for COVID, right? And a lot of that work was done, some of it was done using these models, right? But some of the other work was done, in wet labs, where you basically said, hey, you know what, I'm going to give this and let's see what happens. Yeah. And based on very, very small samples, yeah, not enough data, people started to jump to the conclusion that some of these drugs will also work in new disease areas, right? So that is what we're talking about. But obviously, when large pharma companies want to do this, they want to do this at scale. They want to do this knowing fully well that this is going to really work, because they have to go and get approvals for these new indications, right? And so, the literature search at that point, the past data that is collected, the models are a lot more robust, because you're feeding it a lot more, let us say, valid data, yeah, or data from different sources, that's not going to bias the results one way or the other. Does it make sense, Rajan?
Fine. Yeah, very good.
Good. All right. So this is the part on discovery, and I'm quickly going to move to another one, which is around PKPD modeling. Yeah. So PKPD is basically pharmacokinetic, pharmacodynamic, right? And we almost have this sort of a carousel in some sense, right? We want to design an experiment. So today, what happens is with PKPD modeling is that we start doing this on animals, right? So we inject our animals with certain doses of a particular drug, right? And then with pharmacokinetics, we try to see, what is it that the body does to the drug, right? How much of it is absorbed? How much of it is metabolized? How much of it is distributed? How much of it is excreted? What is the, time of maximum concentration? What is the maximum concentration itself, right? So if I give somebody 10 milligrams of a drug, are they getting 10 milligrams? Are they getting 9? Are they getting 8? Are they getting 7? Is it impacted by other drugs that you're taking? Is it impacted by food that you're taking, and so on, right? And so we do this with large, set of animals. And then we also move this, into humans. But what we want to try and do is to say that while we are doing this, we want to design these experiments and perform them, but we want to take the data that we get and try to start fitting models so that we don't need to have as many animal samples, for example. We will be doing much better in terms of starting at the right dose in humans. I mean, today, the way we do this is that we actually get a drug, a particular dose level, we call LC50. We get that, or LD50, we get that from an animal, right? From animal models. And then we start at 1/100th of that dose, just to make sure that we don't, expose humans to really toxic levels of the dose of the drug. But by starting that so low, what you have to do is you end up giving a lot of people, a lot of patients, ineffective levels of that dose, till you get to a point where the drug starts to become effective, because you give it the right dose. And so if our models were better, or the fitting of our models were better, then in that sense, we would actually be able to move our drug development journey that much faster, right? So we analyze the output, we create another tentative model, and then we go back again to designing our next set of experiments and so on. Yeah. So to give you an example, this is an application of modeling to population PK models, right? So we start with observations or data, okay? We estimate the parameters, we create a model structure, right? Based on that, we go back and collect more data, right? At some point from here, we start to get new experimental data. And then we get the new experimental data, we start to re-estimate the parameters and recreate the model, and then keep on going around, right? there is a component here of expert knowledge, because at some point we can't base it only on models. At some point, we're going to have a scientist coming in and saying, I don't think that anything beyond 100 milligrams of this drug is going to work. So no point in going beyond that. Or I don't think there is any point in starting at 1 because we're not going to see any efficacy or any effect of the drug below 50 milligrams or whatever it is, right? So we have that component of expert modeling, but this is really an application of how we use modeling when we have to talk about population PK parameters. Yeah, and what we finally get is output such as these. So basically, this is your time. Let us say this is your concentration, and you start to say for different doses of the drug, what is the concentration profile? How can I model this, or do I actually need to collect all of these data points? Can I reduce the number of data points that I collect? Because imagine each and every data point is a draw of blood from a human patient. So can I start to do this lesser and lesser because my models are better? Yeah. So that is the second one. And I know we're running out of time. So I'm quickly going to jump to the last one, and then I'll open it up again for questions, which is, so we started with discovery, where we said, how do we get new candidate drugs or molecules? We moved to PKPD, where we talked about, how can we estimate our dose selection, which you find here as well. And then finally, in the development phase, we want to try and find out how can we get supportive evidence for efficacy, for safety? How can I use modeling to inform my clinical trial design? Yeah. And so this is sort of a framework that we use. Yeah. So we formulate a problem. Yeah. And then we figure out whether is it an observational experiment, is it experimental, or am I going to model it? Right. And based on that, what is going to be my model design? Yeah. And if any of these, how do I go around and create what I call a statistical analysis plan? If it is experimental, then I need a plan. I need to figure out what my power of the study is, and so on and so forth, right? But if I am going to design this study as a modeling experiment, then I need to figure out what is my mathematical representation, what is my input data, because your input data, all of us do Wordle these days, right? So we know the starting values are really important. So what is going to be my input data and my starting values? What are the parameters? How am I going to estimate these parameters? How am I going to implement this in silico? And then finally, how am I going to evaluate my predictions, my sensitivity, my specificity, my uncertainty, and so on and so forth, right? So this is the approach, the framework that we normally use when we say, can I do a simulation-based approach to figuring out what could be potential efficacy, safety concerns, what could be a potential design for the study in the development phase. Yeah, so I think those were, I had to rush through the last couple of slides, but I'm happy to stop here and see if there are any questions that I can take.
So, Dr. Mahesh, hi.
There is one question. Oh, sorry. There's one question. This is regarding 3D modeling. So in drug discovery, particularly in omics, what would be, the vital thing you would look in 3D modeling?
So, so in 3D modeling, the biggest thing that we look at is, so I don't want to get into why it doesn't work, because see, what happens in a 3D model is you're modeling the molecular structure, right? And then you're modeling, let's say if there is a drug or if there's a disease area, you're modeling where the spike proteins are going to be. You're going to model where the drug will come and dock on that particular molecule, right? And what is the strength of that ligand, right? So this is kind of what you're going to model. Unfortunately, what happens is that in real life, a lot of this is very, very, it's not set in stone, for example. Yeah. So there's a lot of, quantum physics, that basically can, change, for example, the charge of an electron, or can change the strength of a particular association of a bond, in the 3D structure. And so that's where today a lot of the effort is going in to say, how can we build a certain level of uncertainty towards the strength, to the valency of these bonds, right? To the strengths of these bonds. How can we build some of these sorts of uncertainties into the model itself? and still be able to predict reasonably well, how well the drug candidate is going to bind to the particular protein or that we want to, right? So that's the big part, and that's where a lot of the current research is ongoing. Is somehow taking into account that uncertainty, that ephemeral nature of changing of valencies and strengths.
Thank you. That was a very lucid presentation. I'm sure the audience would have found it very interesting, particularly that thing about text mining, which I think should also, evolve into collaboration between various companies. And of course, the structural functional relationship, which is of the highest importance. And I'm happy that you touched upon PKPD modeling, because that is where I find many of the startups stumbling.
Yeah.
And yes, and just for the information of the audience, in a contest last year on drug discovery, how to accelerate drug discovery, an IT company, a software company, walked away with the first prize. So that should give impetus to the people who are experts in, in software or IT development, software development. Thank you very much, Mahesh.
Thanks,
And we look
Manoj.
forward to it.
Thanks, Manoj.
Yeah.
Thank you.
And should there be any more questions, we'll get back to you.
Sure.
Yeah, I have a question. So I was actually part of the QSAR team about 11 years back at Strand Life Sciences. One of the questions I always had is how soon do you think this physics model is going to come about, which will be more accurate? Is there any open source team, or is there an open source platform that's working on this? And since we have compute power in surplus today, and we did not have compute power in surplus back when I was in this space, when do you see that all possible targets to all possible molecules to all possible variations on the quantum physics front, that it can be simulated, and therefore you will be able to predict the drug as soon as you see a certain condition or a new spike protein, for instance?
Yeah. So Vikram, I have a very, as a very short answer to that, excellent question. I still see us a little ways off. I'm afraid that we're not really, at least, most of the time I tend to see the glass half full, but in this particular case, I see us really trying to simplify the problem far too much and not dealing with it with the level of complexity that it deserves most of the time, right? And so I think that we still are a little ways off. Unfortunately, most of the companies that start off as open source platforms end up, being absorbed or subsumed by the larger pharma companies, and so therefore, they start to lose that collaborative edge. So I think what needs to happen is the reverse needs to happen. There is one example of Janssen now, having a particular, group within their company, which they're trying to spin off as an independent entity so that they can actually be more collaborative and get more things coming in, right? So I think that's what needs to happen, because today, there are like 2 or 3 examples of companies that started off wanting to build open platforms, for this sort of collaboration, so that, because that is absolutely required. One company can't do this by themselves. But most of the time, 3-4 years into their journey, they end up getting absorbed by one of the larger pharma companies, and then they sort of lose their potency.
Thank you.
There are more questions on real-world evidence and real-world data, but we take them up separately.
Done. Thank you. Thank you.
Thank you, Mahesh. Thank you very much for your lucid and we look forward to interacting with you in future. Thank you.
Okay. Ranajit, I would request you to invite the next speaker.
Vikraman?
Hi, Ranajit.
It is now my privilege. Yes.
Yes, please go ahead.
It is now my privileged intent to invite Dr. Satya Lakshmi to give her talk on digital transformation in pharma and biotech. Dr. Satya Lakshmi holds a PhD in molecular biology from CCMB and has worked across the drug discovery value chain in organizations in India for over a decade now. Dr. Satya Lakshmi has extensive experience building and deploying informatics, software solutions, and engages with pharma biotech research management in providing consulting services on digital transformation. She also holds a certificate in business excellence from Columbia University, New York, and is known to collaborate with customers across the globe in building informatics solutions for improved research outcomes. She applies drug discovery and development experience with software knowledge to speed up efficiency and productivity of research labs through digital technologies. Over to you, Dr. Satya Lakshmi.
Thank you, Mr. Ranajit, and thanks to the IKP team for giving me this opportunity. I hope I'm audible, and I would like to share my screen to start the presentation. Let me know when you start seeing my screen. Thank you for the introduction and this opportunity. I hope you can see my screen.
Yes, we can see your screen.
Yeah, thank you so much. So I represent actually IDBS. IDBS is into the research informatics software solutions provider since almost 3 decades, and it's a pleasure to work with such an organization. And what I would like to talk to you today is about the digital transformation of pharma R&D. And when I say digital transformation, we hear this too often nowadays. And when I say digital transformation, I'm talking about an organization of pharma R&D that is giving me for the just before I start off, I think I would just like to switch off my video in case somebody has any bandwidth issues. So just give me 1 second while I do that.
Your video is off, ma'am.
Okay, great. So I hope you can see my screen now again. Perfect.
Yes, we can.
So the drug discovery industry, what Dr. Iyer talked about previously in the previous discussion was more around when we look at the problem statement from an individual drug development point of view. But when you talk about a pharma biotech company, they are developing many drugs across the pipeline, various stages, many scientists involved. So there is a huge amount of data that is getting generated. So the problem statement that I'm going to talk about today is around the data management that comes along with it, the challenges, and what we are doing. Towards the end, I would also give you a snapshot of what IDBS would be doing in that as an example of what is possible in this space and what is the need for solutions. So when I say drug discovery and development hurdles, this is something that gives you a brief idea about what the scenario looks like. For example, what is the attrition rate, meaning what you start off with and what you end with, and how much time does it take to cross each of these stages. And when I say one stage, that could involve many people working in a single lab across sites, across continents, across multiple labs, etc. So that's where the whole data problem becomes manifold. And also we need to combine the data from this stage with somewhere around this stage, go back and forth. And those are the things that actually create that volume of the volume of the data that is generated and the variety of data that is generated, tackling both at the same time so that we are getting this one drug molecule, one wonder drug, which actually goes to the patients. So that's the problem statement that we're talking about. And why is it so huge? because what we are seeing from a company point of view, so I'm going from one individual drug level to a company level or a pharma biotech R&D organization level, where a scientist or a group of scientists are working on multiple projects at the same time. When you have groups of labs and experiments, what you see here are the activities that they do and what they need to do on a daily basis. That involves people, and there is a fair bit of process added to it. It is not a standalone lab where you can probably change the process by looking at the results, etc. So there is a set process that need to be followed. There are approvals, there are sign-offs, etc., all kicking in into the system. You also have a lot of moving entities in a laboratory, which are the reagents, the equipments, or the samples that need all these. These are all the management. This is the management that needs to be done on those moving entities. Then you need to combine the data that is coming from instruments. If we see the lab today, it is coming from a large amount of the data is coming from the machines, and only a small amount of data is manual. So there is a combination that needs to happen, that we need to build the context of the data into the machine. We need to have seamless ways of transferring that information from a machine into the lab notebook, which gives you the transcription errors, which gives you the data integrity problems, etc. Finally, the compilation and analysis is where you decide the speed and accuracy of your programs, and that's what decides the velocity of drug development. Now, what does it come to? All this can be broken down into, there are scientific processes and data management that needs to happen. Either you are a biological entity organization or you're creating chemical entities. There are inter-lab processes communication that happens. You need to streamline and structure all that, because you need to be able to identify the bottlenecks so that you can work on the bottlenecks to remove those bottlenecks and move the programs forward. So probably in a particular program A, there is a bottleneck at analytical data coming faster, because the drug molecule in question or being developed is very difficult. In another case, the example could include something where the drug program is not moving forward because the data compilation is happening very slow. So there are different reasons for every program being stalled. So those are the things that you need to understand through these communications. And finally, that is what improves your efficiency and productivity when you look at it from an organizational level. Because for the top management, all the programs are equally critical, and they need to be able to move all those programs ultimately to a commercial success and a regulatory approval. So whatever we have talked about so far, is it the problem of an organization can be broken down into parts, like what we discussed in the previous session? Maybe that's a scientific problem, or that's a problem where science is getting involved, where you're collecting data and you're trying to understand the data in a much better way and trying to give answers, much better answers going forward. But a lot of this problem that I'm talking about in this presentation is on the data management. So when I say data management, there's a survey that was taken up, which has given you this results, which says that why exactly are the data, what are the data challenges that the pharma life sciences or pharma biotech industry is facing. Number one, the data is scattered all across multiple repositories, multiple places. This is creating a lot of silos in the organization where there is no data flow. There are stagnant, the data is stagnant wherever it is generated. It is not moving as fast as it should move to its destination or where it is needed. Then there is no visibility to the access or visibility to the data, and there is not access as in when you want the data or at the time you want the data. There are delays in accessing the data, or there is a lot of dependency on the IT teams to give the data. And when the data is finally arriving, for you to be able to compile, that becomes a huge challenge. Then changing regulations, obviously, this is a very a simple problem statement to understand. how regulations actually keep changing the parameters so that the data analysis and the outcome, the interpretations have to be adjusted accordingly. We also have seen that the the big pharma industry is now split into a lot of collaborative ecosystem with the smaller spheres of collaboration partners. So this is all very happening through PowerPoint presentation where you're sharing the compiled data. But what I'm seeing in a PowerPoint presentation is actually an interpretation that somebody else is putting onto the data. So if there is a need to look at it from an independent point of view, that raw data needs to be shared, then that data again needs to be prepared. So there is a lot of informal and insecure data sharing that is happening. Then again, there are lots of other issues which actually boil down to these 4 parameters: the data assembly, the data access, integrity, and the quality statements. So when it comes to data, a lot of the drug discovery development velocity in the pharma, biopharma, R&D can be brought down with by addressing these challenges. So I'll just take a pause here and check if there are any questions. Otherwise, I'll move on to the next slide. So I'm hoping there are no questions.
I think we take up the questions after you finish your presentation, because
Perfect.
so far, the attention is all glued to your screen.
Okay,
so
perfect.
I suppose the people.
Thanks. So yeah, so what I was discussing so far, what I have been trying to share with you all is that when there is research happening in a lab, the data management problem is limited only to the assembly component of it, because you have 100% access to the data that you want and whenever you want, because you are the owner of the data. But when you go to a large pharma or R&D organization, the ownership changes as the data takes its journey forward. And that creates the access problems. The access problems is also created because of the different software systems that you use to manage the data. What is generated out of a spectrophotometer? What is generated out of a software versus what is done on the lab bench? All these pieces of data, we all know that how do we if we break down the problem statement into what we see as how do if a large amount of my data is sitting in Excel, and if I have to present it in a PPT, I need to aggregate it, draw out a graph, and then paste it in my PPT and then take it forward. This looks fairly simple because I have access to Excel, I have access to PowerPoint, I have access to all the drawing tools that are available in Excel. But when it comes to multiple different systems which have access, there are licenses involved, there is logins involved. Some people have the access to certain software, some people have certain access to other software. Everybody has their lab notebook and there is dependency created. So there are lots of access issues that get created that needs to be resolved as ASAPs, because that access problem will slow down your insight piece and the final output of your research program. So data integrity is a very important component. We all know that how much data integrity is important. Any faulty or any willful or unwillful data integrity issue can have very serious repercussions for the organizations. And there are lots of audits that are performed, and getting data from this one silo and bridging it to the other silo most often involves a manual step where it could be prone to errors. So this is a very important piece where there is a lot of software organizations working to bridge that. For example, how do you get data automatically from an instrument into a software? That is where we are saying that there is no manual intervention and there is no way anybody can create an error while transcribing the data, or even willfully manipulating the data. Finally, what we are talking about is data quality. After all these steps, what is the quality of data that I'm getting? Is it ready to be consumed? Is it ready for giving me the insights that I want to get out of the data and move the program to the next level, or at least help me take a go no go decision. That right now, if the data quality is not good, we would not be able to reach that insight piece, and we would not be able to make that decision. So just to give you a slight perspective on what exactly we have all talked about data management. It's a very big buzzword, and data is the new currency or the new oil or whatever way you want to call it. But if we try to really break down the problem into two simple pillars of what exactly is management, the dictionary definition of management means the ability to control or organize anything. So what we need to be able to control the data in a pharma biotech R&D is how you record the data. Every single scientist should be able to record that. Similar, if you're doing an ELISA experiment or a spectrophotometer experiment or a CD experiment or any of these experiments, they should be able to record it in the same way. Whatever is getting made in the lab, it should be available to everyone, because that would avoid a lot of wastage and rework burden on the lab. And what is the output? As I already talked about, this is the most important piece where we need to automate the transcription. The process flow is what needs to be organized. Who does what happens when, so that people are clear that once this is not working or this specification limit is not reached, this is where I need to go back, because this back and forth is where this ambiguity is what wastes a lot of time. And then by the time you go back to the experiment, you already lose a week of time or a 10 days of time. By the time you assemble the data, analyze it, and then take a decision and go back to another level of experiment or another change in the experiment design. The layers, where do you need to find information when we're talking about information? Right now, if the information is all residing in papers, there is no way we can search for the information. So we need to be able to add that hierarchy of data project. Under that, what are the objectives under which I'm creating that data? So this is what will help you organize all that data. Then at the final, how do we align all this data with some unique IDs? So if every experiment that I'm doing has a unique ID, every sample that I'm generating has a unique ID, there is a very good possibility of being able to search that piece of entity from the system and align everything else that has happened on that unique ID. Wherever I'm using that unique ID across experiments, if I've used that unique ID of sample across 10 analytical experiments, I will be able to pull out all the 10 experiments just by looking for that sample ID. So that's the way you need to align diverse pieces of data together. Just to give you a perspective of what is the data lifecycle that goes around. So in an organization, there is creation of data, there is processing, and there are various needs that the data goes through as it completes its journey. So in a non-digital system, this process is very ambiguous, in meaning it may be going from a create to a use step from by only one person, whereas in a digital system, everybody would have access to this process, use, transfer, store, share, archive, etc. And there is no destruction of data because everything is archived and everything is traceable and everything is audit available. So there is a lot of a benefit of going digital when you talk about the various steps of the data lifecycle, and this becomes very important. As I said, I'm re-emphasizing that for a single lab doing agile experimentation, this may not look like a big problem statement, but imagine a pharma biotech R&D with hundreds of scientists spread all across the organization, across various levels of hierarchy, doing different types of experimentation. This organizing and streamlining all this becomes very, very crucial and critical for the management to see that the program has made progress across 3 months or 6 months or 1 year or whatever is the timeline they have set for the project to succeed. Just to say, give you a way of how it happens. When you work in a non-digital system, this is how your protocol looks. Very, very simple, that I just need to record whatever I'm doing. I have complete freedom to write wherever pieces of data fit in. if you look at it from a data perspective, if you wear the data hat and look at it, you would be able to recognize that this has volume data, this has time data, this has text data, this has number data, this has units data, this has a graphical output, this has a tabular output. So this is how you need to wear the data hat and then analyze, and that is when you will be able to streamline into these kind of settings, where you see that first I need to write what is the context of my data, then I need to be able to capture what exactly is my data. Is it semi-manual? It's manual. Do I just need a table, or do I need this table to be filled automatically through a software? What is it that I need to do? As soon as I fill this table, this graph should appear automatically. So these are some of the things that can be automated. So when I talk about automation of R&D, this is where it can become very easy, so that the scientists.
Sorry for interrupting, Dr. Satyalakshmi. Can you wrap up in a minute?
Sure, sure. So like I said, this is the data integrity question, and very important for all the pharma R&D is your data integrity deficiencies that are addressed by these Alcoa Plus principles, where the data needs to be in all these formats. It needs to be attributable, legible, etc. Data quality, it's very important, and what you need to see here is that the data that you're generating, is it unique, accurate, or complete for use across its life cycle. And unless until your data is having all these attributes, it would not be of a high quality, and it may have to be reworked and redone again. Then is data the buzzword, or is it really a critical factor? Yes. So there is a measurement that has been done, and it is identified, it has been identified that the bad data quality does have an impact on the life sciences company output. And when you have, what you need to do to have digital success is to manage your data operational efficiency that we talked about, and reduce the speed of getting the drug to the clinical trials, and also manage all your moving items of material samples, etc. So with that, I just want to close with this slide, which is from IDBS. IDBS does this kind of system for biopharma lifecycle management, where we are trying to automate all these different pieces with our Polar platform by integrating the workflows and the insights and everything, so that there can be a lot of accelerated therapeutics that can happen. With that, I just would like to wrap up, and over to you, Ranajit.
Thank you, Dr. Satyalakshmi, for for evangelizing the data management part of digital transformation in R&D. And there are certain things which I think which the applicant should note, is that data process is critical. And another thing which you highlighted, and there is a question on that, in terms of data in data integrity, how important is computer systems validation?
Yeah.
In a GX environment.
Correct. It's a great question, and it is very, very important and critical for all the pharma biotech R&D, because as I said, the data is the data where it gets created versus where it gets used. They are very far apart. And to maintain the data integrity, and when you are using computer systems, it also needs to be regulated so that nobody can touch the system and nobody can tamper with the system. So the computer systems validation is a very important component of the FDA 21 CFR regulations. And any software system, there is a level which is called as GAMP levels, and any software that comes into the organization will be undergoing the computer systems validation. There is a IQ, OQ, PQ performed on the system, and there are access controls that need to be given, and there are usernames and passwords that should not be shared across users. There are lots of guidelines that are set by the FDA, and that all software providers need to provide in their systems.
Thank you, Dr. Satyalakshmi. Many of the startups do not recognize the importance of regulatory systems. So your process flow has to be regulated. That is one thing which is a takeaway for the startups. Thank you very much again, Dr.
Thank you.
We now move on to the next speaker, uh, Dr. Sridevi Challa. She is a chemical engineer by educational background, master black belt, and a certified Scrum Master. Sridevi is an experienced process engineer and an efficient people manager with versatile experience in academia as well as the pharmaceutical industry, ranging from APIs, which is active pharma ingredients, oral solid dosages, complex generics like microspheres, liposomes, and ophthalmic suspensions in nanoemulsions. She is currently team lead formulation development at Sandoz, which is the generics arm of Novartis. And in addition to formulation development, she also drives the QBD, which is quality by design, data science and digital capability building for the formulation development team. She is an active researcher pursuing her doctoral at IIT Bombay on the academic front and is actively involved driving several digital and OpEx initiatives in the organization. She has worked closely with startups through Novartis Bio platform and delivered successful POCs that is now being scaled across Sandoz and Novartis. Over to you, Dr. Sridevi.
Thank you so much, Dr. Ranajit Singh, for the introduction. I hope I am audible. Let me just share my screen. I hope my screen is visible to all of you.
Yes, your screen is visible.
Thank you. Here, a little disclaimer. This presentation is based on publicly available information. These slides are intended for educational purposes only and for the personal use of the audience. These slides are not intended for wider distribution outside the intended purpose without presenter approval. The content of this slide deck is accurate to the best of presenter's knowledge at the time of production. All right, so this is about the disclaimer. I'll just go off video just to enable bandwidth. I hope that's fine. It might be stating the obvious today after the being a speaker following Dr. Mahesh Iyer and Dr. Satyalakshmi Oruganti. But I would still state it because the session demands it, or let's put it this way, that this is the theme of the session. Okay. So we stand on the brink of a technological revolution that will fundamentally alter the way we work, we live, and relate to one another. And in its scale, scope, and complexity, the rate at which this transformation is happening today is unlike anything that mankind has witnessed or experienced before. On that note, I would like to draw your attention quickly to the journey we have had so far on the roadmap of our industrial revolution. The first era of industrial revolution, or popularly known as the Industrial Revolution 1.0, earmarked for its usage of water and steam to mechanize production. The second one used electric power to create mass production. And the third industrial revolution used electronics and information technology to automate production. Now it is the time for the fourth industrial revolution, popularly known as Industry 4.0, aka Pharma 4.0 in the pharmaceutical industry. And this is the digital revolution. It began since the middle of last century. And the digital revolution is characterized by a fusion of technologies that is blurring the lines between the physical, biological, and the digital spheres. On that note, today's talk, I want to focus on data-driven digital twins, the vision in pharma and biopharma R&D and manufacturing. Outline for today's talk goes like this. I'll just quickly go through what is a digital twin, the need for a digital twin in the pharmaceutical industry, examples of typical unit operations in the pharma, biopharma, and expectations from digital twin, and a few possible conclusions or key takeaways from today's session. All right, so what is a digital twin? As you can see here, so digital twins are virtual constructs of physical systems that mirror the behavior of dynamics of these systems. They can be digital twins for products, processes, or equipment. Basically, digital twin is a virtual representation of a physical product, a process which can be used to understand and predict the physical counterpart's performance characteristics. And these digital twins enable us to perform experiments, or to put it in layman's terms, they enable us to perform the experiments on the computer, also known as the in silico experimentation, before we actually invest in the physical assets. And so we can actually see what is the impact of various parameters, critical process parameters on our CQAs, and then we can optimize the process virtually by performing these in silico experimentation and finally arrive at the best possible optimized parameters for the process using these digital twins. So today I'll be focusing mostly on the process digital twins and the need for these in the pharmaceutical industry. So if you look at the digital twins in pharma or biopharma, so there are a lot of parallel unit operations happening. And if you look at the way these digital twins can be put, so there are like simple twins, computational twins, machine learning based twins. Some are very complex twins and they also interact with the PAT tools and also provide some real-time analysis as well as control of the process that is happening at the production scale as well. Right? Now, when you look at the process digital twins in the pharma or biopharma particularly, so I'm not sure about the background of today's audience, but I'm certain that all of you have seen a tablet or a syrup or an injection or something or the other through, right? You might have come across. And if you have noticed the label of these, you must have seen, for let's say you take a tablet. If you look at the label claim, there are like multiple ingredients in that tablet, like a lot of excipients. They could be binders, filling agents, colorants, taste masking agents, along with the active pharmaceutical ingredient. So let's say if you imagine the end product as your tablet, then to get to the tablet, the initial ingredients are a lot of these raw materials like the different excipients, which could be binders, fillers, so on and so forth, and the active pharmaceutical ingredient. So if you start with that as your step one, by the time you get to the final tablets, what you have is a whole chain or a number of steps of processes involved, for like sieving, blending, roller compaction, high shear granulation, or compression, tablet coating, so on and so forth, depending on the type of the final oral dosage, final solid dosage form, or injectable, or a liquid solution, or whichever is the pharmaceutical medicine of interest that you are producing. So what I would say is that the point here is that there are multiple unit operations involved, and by the time we meet the final product and the CQA, it is important that at each and every step throughout this equipment train, you need to have the right process parameters, the right ingredients from one step to the other step, so that you get the final product with the desired or the target critical quality attributes. So this is where a lot of the material properties, process conditions, which are the critical process parameters, and the equipment characteristics, all these become inputs to these process digital twins. And each unit operation involved in this train of pharmaceutical unit operations will have one process digital twin, and then that will help us enable and predict what will happen if I do the experiment at this particular condition or set of process parameters and the formulation composition. Now, if you look at that about the basics of the pharmaceutical, the flow of how the pharmaceutical drug is manufactured. And now when it comes to the architecture in leveraging the digital twin, you can see there are multiple inputs that are required. There is some equipment related characteristics at different scales. For example, you might have optimized a process at the laboratory scale and you want to transfer the process to a plant scale. A lot of the times you might not be even having same make and model of the equipment which you used at lab scale at the R&D scale. It might be a different make, a different scale, etc. So all these scale dependent parameters, the different types of equipment that are used, and there could be a lot of the other material related parameters. So all these will become inputs to the process digital twin. And inside the digital twin, what will be there is there could be quick guidance formulae based on first principles, or there could be some complex digital twins which could be based on discrete element modeling type of framework, or a finite element modeling type of framework, or a population balance modeling. So again, there are multiple different modeling and simulation methods that are available, and not one solution fits all the unit operations. And in addition to all these, there are also now these machine learning twins where even without having this entire understanding of the underlying physics, if you have a number of trials done already on a particular machine with similar molecules, then what would happen to my next trial on a similar machine can be predicted if we already have a machine learning based twin. So all these will help in making the right decision, and instead of taking trials at plant scale and then noticing that, okay, hey, this parameter at this value wouldn't give me the product of this CQA, which if I realize after taking the trial, that would result in wastage of money, as well as a lot of the raw materials that are put into that system already, time of FTE time, etc. So before even doing that, we would be able to run the trial and trying to understand what would happen and what would be the best parameters at production scale, especially. So now here I have put few unit operations. For example, we have a blending step. So what could be the ingredients to the blending digital twin? These are material properties like PSD, flow characteristics, the process conditions, including order level, RPM, the equipment geometry in the form of, say, STL files, if you're using a DEM kind of a model. And then the output that you can expect out of it is at what conditions I can get the blend uniformity, which are my optimal conditions at lab scale first, and what are the conditions at which if I take the trials at plant scale, I would achieve similar CQAs as I have been achieving at the lab scale, like the blend uniformity, COV of uniformity, blend flow characteristics, etc. And one other unit operation which you can think of is I have put a few critical unit operations and what would be the inputs to a process digital twin and what could be the output. So if you take tablet coating as an example, then the material properties could be solvent volatility, viscosity, surface tension, so on and so forth, and process conditions including inlet air, temperature, equipment geometry, and the outputs that it could predict at different combinations of the ingredients on the left-hand side. The digital twin would be able to predict what would happen to my coating quality and the performance. Similarly, for high shear granulation, if you think of a high shear granulation digital twin, there are different material properties, process conditions, equipment geometry, which can be given as inputs, and it would give me, the digital twin would give me the combination of process parameters and the material properties that would allow me to get this target CQAs at different scales and optimized parameters. When it comes to biopharma, there could be some of these operations of interest like chromatography. Again, even here, here I have just put how the model will be in the digital twin for the data-driven modeling, and what are the optimization strategy constraints and some of the modeling equations involved. And here, oftentimes there is minimum yield, minimum purity, and we want to predict the conditions at which I can get maximum yield and maximum purity with minimum column pressure drop. And bioreactors, again, even in bioreactors, how these digital twins will help me. They will help me find out or figure out the conditions that will allow me to get optimized parameters at lab scale for the biomass, target biomass concentration and dissolved oxygen and substrate concentration, as well as how do I scale the process from lab scale to the plant scale. And there are a lot of advanced modeling framework involved in these digital twins for the bioreactors, especially, which involves extensive background, computational and mathematical background, including a knowledge of fluid dynamics, bubble dynamics, mass transfer, transport phenomena, etc. So if you see right now, until now, you have seen a lot of the different unit operations, which or processes, which might be of interest, on which I can pick and make a digital twin. What would be the inputs and what will be the outputs? If you see, if you can notice pick a trend, then you might have noticed that in all these things, all I want to do is, before even doing the experiments or with minimal number of experiments at lab scale, I have two goals. One is, I want to optimize my process at lab scale with minimal number of experiments and enhanced product and process understanding. And I want to transfer the process to this plant scale or the production scale, again, with right first time scale up and without any loss of the time or cost involved in taking multiple batches, avoiding the batch failures and getting the right first time scale up. So this is where these process digital twins will help. And to conclude key takeaways, these digital twins enable users to reduce experimentation and rapidly explore the design space for enhanced process understanding. So this is going to be like a combination of few minimal experiments, screening experiments, instead of 50 experiments or 60 experiments within, let's say, we can try to get similar understanding or even more enhanced understanding of the process with just 10 experiments or 15 experiments in the lab scale. And then once we validate the model, then these models can be used during product development, optimization, scale up, as well as tech transfer stages. And with the help of these digital twins, we'll have enhanced process understanding from the in silico trials, which allow transfer of process from different scales, different types of equipment, and reduced process downtime. And deploying these data-driven digital twins not only enhances process understanding, but it also reduces the cost of product development, manufacturing, and it also accelerates the timeline significantly, which is the need of the hour. So this is what we have been deploying now and trying to use some cutting edge modeling and simulation techniques to develop the digital twins and deploy them for assisting the formulation scientists for accelerated product development. With that, I will now take a pause and see if there are any questions. Thank you.
I received one question on WhatsApp. Quality by design is now a kind of a buzzword in drug development. So how can a digital, how can digital transformation, leveraging digital transformation enable quality by design?
Mm-hmm. Thank you, Ranaji. That's a very interesting question, and this is something like a need of the hour for all of us to know this answer. So, what I can say, based on my knowledge, is that the moment we have a digital twin, it means that we have the process understanding. Even before taking the trial, we can tell what will happen to my process if I change some of the process parameters, let's say a blender RPM higher or lower, or in a coating the spray rate higher or lower. So instead of shooting arrows in the dark, I would be more informed because I have a underlying first principles or the physics captured in my digital twin. So even before taking the trial, I know what would happen, and I would also be proving with few lab scale experiments, which means I have enhanced process understanding as well as product understanding, which is what is all about quality by design. It's not like you have a product, you measure of the CQAs, and then you know that, okay, this is not meeting my specs. That means quality by testing. But quality by design means you already know, you can tell that, okay, this parameter will not lead to the CQAs that I am going to target today. So I'll need to work in this design space. Only then I'll be able to get these CQAs. So this is what is QPD. And I believe these digital twins will enable us to build or develop this kind of a knowledge about the process. I hope I have answered your question.
Yeah, I do think so. Thank you very much, Dr. Sridevi Challa. I know you haven't got your doctorate as yet, but just out of today, the season being Women's Day, so out of respect. So it's a very good overview that you gave of how pharma is evolving into pharma 4.0 and how digital twins can be employed in scale up. In fact, what you presented, if I put it in a nutshell, you have digitized a pharma production unit, if I may say so. And actually, you brought up this, the importance of CPP and CQA, the critical production process parameters and critical quality attributes. These are something which I think our startup should start thinking about. Thank you very much. Thank you very much. We shall stay connected.
Thank you very much. And thanks for the wonderful summary, Ranajit, Dr. Ranajit. Thank you.
Ranajit, I request you to invite the next speaker, final speaker for the day, Mr. S.V.N. Raju.
It's my pleasure now to introduce Dr. S.V.N. Raju, who is the co-founder of Apramita Innovations. Dr. S.V.N. Raju has 27 years of pharma R&D exposure in the areas of formulation development, tech transfer, production, intellectual property management, product selection, and portfolio. S.V.N. Raju has worked with Dr. Reddy Sipla and most recently as SVP R&D at ASEAN Drug Research. He has also been granted 2 US patents and has 20 plus patent applications to his credit. At his venture in Apramita Innovations, S.V.N. Raju aspires to bring cultural change in formulations and analytical teams so that both of them learn other areas while they continue to master their respective fields. Over to you, Dr. S.V.N. Raju.
Hi, good evening all. Thanks for IKP team and Dr. Ranjit sir for your nice introduction. So unlike other speakers, I will be just sharing my experience as a startup as well as during last 30 years of my journey in the pharma industry. What is the importance of data and how the digitalization helps the pharma industry. So you're able to see the screen?
Yes, we are.
Yeah, so about 30 plus years back, I've started my journey in pharma. And since then, I've been hearing whenever there is a presentation or a talk regarding pharma, the first thing is when it comes to the new product development, it's low rate of introductions. Sorry, the pharmaceutical development, particularly the NCEs and NBEs, they need very high investments to tune of $1 to $2 billion. and longer duration. About it takes 12 to 15 years to complete the development. And the probability of success is 1 in 10,000. This is what I've been hearing. And even now, after 30 years, I've not observed any change in these statements. So one reason which we could attribute to this is the broader adaptation of this data management in healthcare is still very, very low. So that's the reason I think it is status quo, the investments are high, longer duration and all that. And when it comes to the Indian pharma companies is yet to get into the market within NCE. Not only that, even Western countries, if we see, for last 60 years, the US FDA has approved only about 1,200 new chemical entities and biologics. That means in a year, approximately 20 new chemical entities only are coming. And when we look at the players, I'm sorry, I think there is some problem with the, so the number of players wise, if we see, only about 6% of companies could register only one ME. And out of these 6% of the companies also, only 12% of the companies are still existing, and remaining companies, 88% of the companies, either they have failed or merged or have been acquired. So the data is very powerful, and to share my experience, the recent experience as a startup, we have started our journey. We have taken up differentiated product development, and we have repurposed one of the existing product into a topical dosage form. And using the available data, we could able to get from the Indian DCGI office, the phase 1 and phase 2 waiver, and directly got the phase 3 approval. So that is the power of data. And the data itself, if it is having that much power, if it is properly used and if it is properly digitalized and made it available to the subsequent companies or the startups, I think we can see the miracles to happen. And whatever the status quo we are talking about, that can be changed. See, in pharma only for last 30 years, as I mentioned, that there is no change in the quantum of work involved or quantum of money required and all that. But if we see last 30 years, the telecom as an example, if we take, there is a sea change in the scenario, right? All of us, we know about 20-30 years back when we were using mobile phone and all that, we are very scared to use, right? If I have to talk to my parents, every minute counts, and every minute I have to pay 20 rupees, 30 rupees. Whereas now it is free. So that's the kind of transformation happened in the telecom industry. And with proper data management and data digitalization, even in pharma industry, the similar kind of thing can happen. And what is the need? See, today, it is my perspective. Today, the pharma scientists or the chemistry scientists, they are having the core knowledge in their core subjects. But unfortunately, they are not acquainted with the data modeling and data handling. So universities have to start looking at modifying the course curriculum to introduce the data modeling tools into the pharma chemistry courses, or even integrated courses would help. And today, the researchers, they are not embracing the role of data scientists. So I might be very good in formulation, but if I don't know how to manage the data or how to interpret the data, then I might not do a good job. So as a formulation scientist, I also have to embrace the role of data scientist. And then I think it can help a lot, and we can bring the products much faster into the market with minimum data. And even regulatory bodies are the publishers, they should start pushing or encouraging either the applicants or the others to submit the raw data in a processable formats, so that it will be easy for the startups who are taking up this journey of digitalization. This data is available, and then it can be easily processable, and we can use. And in my experience, I've seen most of the companies, they begin their journey of starting a data modeling or with an intent, they start that program. But unfortunately, after 2 years, 3 years, 4 years, that program becomes just as a database, because this data modeling requires a lot of data to be pooled and fed to the system. By the time this happens, the people might change, or companies' priorities might change. With all this, what we see is whatever systems they have built in, they just become database, and the processing part is lost. I think if academics as well as the industry focuses on this, and even startups can benefit from this kind of a data collection as the course curriculum changes, these startups are the new entrants into the industry, can benefit and do a good job in managing the total show. Thank you.
Thank you, Dr. Raju, for enlightening us on your journey and how you managed to bring a formulation onto the market, and the importance of submitting data to accelerate your formulation development. Thank you very much. We unfortunately do not have time for questions, so I hand it over to Vikraman to round up the proceedings.
Thank you.
Thank you, Ranajit. And a big thank you to all of the speakers who took the time out to join us today. Dr. Mahesh Iyer, Dr. Satyalakshmi Oruganti, Ms. Sridevi Challa, and Mr. S.V.N. Raju, we thank you so much for coming and being part of this Amrit Grand Challenge Jan Care webinar. To all of the participants, please do visit the BIRAC website and submit your applications before March 31st. We hope to stay in touch and to hear from you regarding your application. Again, thank you, Ranajit, and thanks to all the speakers for this wonderful webinar. Please do stay in touch. We'll keep you posted on the progress of the Grand Challenge program. Dr. Vishu, do we have any other announcements to make to the participants?
No, Vikraman, that's it.
All right. Thank you, everybody, and please stay tuned. We will plan a few more webinars. Though this was the last one as part of this series, we will have a few more webinars, so to stay in touch, and we will keep you updated. Thank you.
Thank you.
Thank you. Thanks a lot.